And now we're going to have an introduction of our speaker by our Cardiovascular Center chairs.
Good morning, everyone. Thank you so much for joining us today for our Speaker Science Seminar Series. My name is Vanya, and I'm the undergraduate co-chair for Center for Cardiovascular Investigations, along with you and Mallory, who is our graduate co-chair. Today we are joined by Dr. Howard Leong-Poi, a clinician scientist and cardiologist and echocardiographer. He is also the medical director for the Heart, Lung, and Vascular program at St. Michael's Hospital.
Dr. Leong-Poi is a professor in the Department of Medicine at the University of Toronto. He completed undergraduate and graduate medical training at the University of Toronto and a research fellowship at the University of Virginia. His primary research focus is on ultrasound-targeted gene and cell-based regenerative therapies for cardiovascular diseases.
He has published over 160 articles in peer-reviewed journals, with his research being funded by the Canadian Institutes of Health Research, the Heart and Stroke Foundation of Canada, and also the Canadian Foundation for Innovation. Please give a warm welcome to Dr. Howard.
So this is the one that's going to amplify out there. And then I'll come back up at the end of your talk.
Okay, good morning, everyone. First of all, I want to thank Dr. Martino and her entire group here at the University of Guelph for inviting me to speak today and to visit the University of Guelph. And I've had a fantastic visit so far, and thanks for the kind introduction. Dr. Martino knows my son goes to the University of Guelph. He's a fourth-year environmental engineering student, and I think he was pleased that I was visiting. He asked me to pick up some groceries on the way in, so I was happy to do so. So, I'm going to talk about some of the research and the work that I've done over the last couple of decades to give you a bit of flavor of what one aspect of cardiovascular research entails, and hopefully to hopefully inspire those of you who are interested in research to pursue it, because it's been quite a remarkable journey for me, and I'm sure those who do research can speak to their own individual supervisors and mentors, and it's really the same all around. It's been a fantastic journey, and one for me that I plan to continue for a little bit longer. So these are my objectives for the talk. I'm going to share a bit of my personal journey. I'm going to introduce you to microbubbles, which is the basis of the research that I do, and one of the aspects that I like about the research that I do, it actually ties into well with my clinical work. I'm a clinician scientist, as you've heard. Some of the basic aspects, the current use of these microbubbles in clinical cardiology, introduce you to the work that I do with regards to ultrasound-mediated gene delivery, which is also called UTMD, or ultrasound-targeted microbubble destruction, a term that was generated back in the early days of this field of research. And some of the applications for this therapies in two major areas. One, in pro-angiogenic therapy for peripheral arterial disease or ischemic peripheral arterial disease, and targeting ischemia reperfusion injury in the heart, which is targeting acute myocardial infarction. And then talk a little bit about some of the challenges to think about translating this to the clinic. So for me, this is where it all started. You heard in your introduction where I did my research fellowship. This is the University of Virginia. And prior to this, I'd gone through clinical cardiology training at the University of Toronto. And I was interested. I always wanted to be a clinical cardiologist. But I had this opportunity to do a fellowship in the US. This is Cobb Hall at UVA. I'm not sure if anybody ever visited UVA. It's kind of similar to Guelph, actually, a small college town. It's beautiful, actually. It's in the Blue Ridge Mountains, just about 2 hours south of DC. It's I kind of think of it as a cultural hub of Virginia, because it's kind of creeping into the south. But very, very international student population. And it's the university that Thomas Jefferson, one of the early presidents of the US, built. So lots of history. So Cobb Hall was where the lab that I went to was housed. You can see it's quite grand. And the names on the slide, Sanjeev Kaul was the main investigator, scientist, who also a clinician scientist. But he had hired 2 young clinician scientists who was part of his group, Kevin Way and Jonathan Lindner, and this group formed the core of the research. And back in the day, this was sort of the center of the universe for microbubble research. It was really emerging technology within the field of cardiology and ultrasound and echocardiography. So what are these microbubbles? It's a contrast agent that we use clinically every day in the echo lab and actually in many ultrasound labs as well. By the term, they're tiny bubbles. They're about 2 to 4 microns in diameter. So they're larger than nanoparticles, for example, but they're smaller than red cells. And because of that, they can traverse through the circulation unimpeded after an intravenous injection. They consist of a shell that has various compositions. It could be lipid, it could be protein-based, and it has an inner gas that could be nitrogen-based. And the newer generation microbubble agents have these high molecular weight perfluorocarbons that allow these microbubbles to persist for several minutes, up to 5-10 minutes in the circulation after an intravenous injection. This is intravital microscopy. This is a cremaster muscle prep. I was talking to Dr. Morant last night, whose lab uses intravital microscopy. And this is fluorescently labeled microbubbles, these bright dots that after an intravenous injection into this mouse, you can see flowing freely through this arteriole. You can see the microbubbles go freely through this capillary bed into this draining venule. And these microbubbles, if you inject them intravenously, they go everywhere that blood goes within the body. These microbubbles are acoustically active, so if you insonify them with ultrasound, they resonate, they oscillate, and they reflect ultrasound that can now be processed by echo or ultrasound systems to create a bright white signal. And from a behavioral flow standpoint, they behave very similar to red cells. And because of that, they can act actually like flow tracers within the body. And that was one of the major applications that was ongoing in the lab when I was there, was looking at blood flow, looking at perfusion with these microbubble contrast agents. And as I mentioned before, we use these agents clinically in the echo lab where I do my clinical work pretty much every day. About anywhere from 25 to 30% of patients get these ultrasound contrast agents. I'm not sure if anybody here has ever had an echocardiogram performed on them. If not, I'm sure everybody here has had a family member who's had an echo performed on them. If you see a cardiologist in today's world, an echocardiogram is one of the standard imaging tests that are used to assess cardiac function, structure, look for valvular abnormalities. And for anybody who's done ultrasound knows that there's limitations for ultrasound in terms of attenuation, in terms of image quality. So you can see this is an echocardiogram or an ultrasound of the heart on the left. These are 2 views of the heart. And in this patient, for technical reasons, for body habitus, lungs, etc., you really can't get very, very good images. And it could result in a potentially non-diagnostic study. So with an intravenous injection of microbubble contrast agents, this is the images shown on the right. It's been colorized. You can see that the contrast agent fills the left ventricular cavity. These are 2 different views of the left ventricle. You can outline the walls of the left ventricle. You can see the endocardium. You can trace them. You can quantify LV systolic function, left ventricular ejection fraction, and you can determine whether or not the patient's left ventricular function is normal, is it abnormal, and have some clues as to why it's abnormal. Is there a prior myocardial infarction, for example? Is this patient at risk for heart failure, for example? And as I said, it's become quite a game changer for us within the echo lab. So during my fellowship, one of the hot areas at that time was, as I mentioned before, using these contrast agents to look at blood flow to the heart. And these are animal studies that were being performed, and then they're already translating this to look at blood flow in humans. And at that time, some of the projects that I worked on and led included looking at flow function relationships in models of acute and chronic coronary disease, so-called myocardial perfusion or MC, myocardial contrast echocardiography, perfusion studies, and we're actually doing some studies in patients in the cath lab, for example, patients who present to the emergency department with chest pain. And the concept is shown on the graph above here. This is a short axis view, open chest animal study, where an occluder has been placed over one of the epicardial coronary arteries to create a stenosis or narrowing to one of the beds in the left circumflex, which is this lateral wall shown here. And after an intravenous injection of these microbubbles, we can examine how these microbubbles replenish within the capillaries in the heart. And if blood flow is normal, as shown in the LED bed here and the yellow line, it replenishes over time and plateaus. But if blood flow is reduced, then that replenishment rate is less and the plateau is maybe a little bit less. And with this, we can actually develop a method to quantify blood flow to the heart. And as I said before, it's been used in clinics to quantify blood flow as a perfusion study. And we've used this in patients. This is an example of left ventricular pacification in a patient with an apical infarct, an apical aneurysm shown here. These are perfusion images here where after destruction of the contrast agent, you can see replenishment within the myocardium here in this apical four-chamber view. And we can use it during stress, whether it's exercise or pharmacologic stress, to look for perfusion deficits. It's a measure, a marker of being able to detect underlying coronary disease in patients. We've actually translated this into the basic science lab, into the animal lab, to look at blood flow in different models. And those examples I've shown on the right. This is a model of hind limb ischemia. This is perfusion imaging in a normal hind limb. I'll let it play again. You can see the flow in the arterioles, arteries, and then slowly into the capillaries as blood flow occurs in the hind limb. This is an ischemic hind limb, so a portion of the artery that feeds this muscle has been ligated and removed. And sorry about that. You can see the reduced blood flow. This is a myocardial perfusion imaging in a rat heart. This is normal perfusion shown here, short axis imaging, replenishment. This is end systolic images. And this is after a ligation of the left anterior descending coronary artery for anybody who does any LED ligation models. You can see the perfusion defect shown here. And this is perfusion of a kidney in a rat. And it could be used to look at renal perfusion as well. So these are applications of these contrast agents that we use in basic biomedical science. Just one application that I'm going to talk about that I did studies doing my fellowship days, but I don't do anymore. This is molecular imaging. And these microbubbles, you can actually attach ligands to them that make them adherent to receptors that are expressed on the endothelial surface of vessels in disease states as a disease detecting imaging modality, so-called molecular imaging. And this is work done during my fellowship on molecular imaging of angiogenesis. This is a mouse cremaster muscle shown here where the microbubbles have ligands that are attached to them and they adhere to these disease state surfaces. This is quantification of attached targeted microbubbles compared to control microbubbles. And this is Matrigel angiogenic plugs that look at the adherence of microbubbles that you detect with ultrasound with two different targeted microbubbles versus isotype control, where you have very little uptake of this contrast agent. And the concept here for targeting angiogenesis was for detection of tumors, so detection of malignancy by imaging modalities, so-called molecular imaging. And during my fellowship, one of the fellows in the lab, Jonathan Christensen, just started working in a new field within microbubbles, and this is the concept of gene delivery, where instead of attaching ligands, you can attach various targets onto these microbubbles, nucleic acids, for example, plasmids, etc. When you inject them intravenously, they go everywhere in the body where blood goes, but you can actually target delivery of these microbubbles and their cargo to specific vascular beds with the external application of ultrasound. So intravenous injection goes wherever blood goes, but with an ultrasound probe, you can target over whatever area you want, whether it's the heart, whether it's skeletal muscle, etc. And this is just intraviolent microscopy, fluorescently labeled DNA on microbubble surface, showing fluorescent DNA that's been deposited within the vascular space and a little bit extravascular. This is scanning EM showing a microporation within the myocytes, so one of the mechanisms for this particular method of gene delivery. So this slide as a schematic of the concept of ultrasound gene delivery, or UTMD, ultrasound-targeted microbubble instruction. So first you have the microbubble nucleic acid complexes, and in our lab we do charge coupling. So we use cationic microbubbles. You have the anionic DNA, whether it's plasmids or microRNAs or other targets. So the target nucleic acid is bound on the surface of these microbubbles. They're injected intravenously. Here in this example, you target ultrasound over the heart, and it leads to targeted delivery, targeted transfection of the myocardium, of the vessels within the myocardium. And a number of studies have looked at the mechanisms for uptake and for transfection, including microjets, sonoporation, and endocytosis. And then, of course, with transfection, you have protein analysis, synthesis, secretion, and biological effect. Sorry, I think that was a bit of feedback. So what are some of the advantages and disadvantages of this technique for gene delivery? I'm sure many of you have heard of viral vectors that can also result in transfection, probably the most commonly known. But some of the advantages of this particular method of gene delivery, first of all, it's non-invasive. There's no injections. It's an intravenous injection of microbubbles and DNA complexes. And because of the non-invasive nature, you can deliver multiple genes, you can deliver multiple deliveries over time. So even if the transfection doesn't last forever, you can still have repeated methods to continue to treat a particular disease state, for example. It's actually very effective as a method for vascular transfection. So if you ever wanted to transfect the endothelial surface, if that was your specific target. And so angiogenesis research, for example, it's quite advantageous for this. And I have a few collaborative projects with other investigators that are looking at targets within the vascular endothelium. And it's actually safe. The nucleic acids that you want to deliver, the transfection rate outside of that ultrasound beam is actually very, very minimal, because the body's own endogenous endonucleases will digest any nucleic acids that are in the bloodstream. So what are some of the disadvantages? Well, first of all, it's a fairly modest transfection efficiency, but that's why you have to give repeated gene deliveries if needed. It can only deliver to organs and cells with an intact microcirculation, because it has to be accessible to ultrasound. So things like bone, for example, lungs, because they're aerated, the amount of transfection in these organs are very minimal to none. And over time, there's been some concern about bioeffects, but just regulation of ultrasound power and frequency, etc. You can actually have some of the bioeffects that result in transfection, but you don't have bioeffects that result in overt tissue destruction. Many different vectors over time, plasmids, minicircles we've looked at. We've done delivery of siRNA and microRNAs. You can also use long non-coding RNAs for anybody who does RNA research. Over time, we've learned that the higher the power, the better the transfection efficiency. You have to be careful about bioeffects, as mentioned. It works via inertial cavitation. Vectors that are bound to bubbles work better than free bubbles. And you need triggered imaging to allow these microbubbles to replenish the organ that you're trying to deliver genes to. So continuous ultrasound does not work as well. And there are ways to modulate the ultrasound to result in extravascular transfection if you want to transfect myocytes, for example, versus transfection of the endothelium. And lots of different disease states. The ones in yellow are the ones that our lab has done at the University of Toronto. So models of peripheral arterial disease, myocardial infarction, dilated cardiomyopathy. We've done some tumor transfection, a lot of work in various labs across the world looking at tumor transfection, and we've also done transfection to large arteries, including the aorta, for targeting abdominal aortic aneurysms. So some of the early work in the lab looked at pro-angiogenic therapy in peripheral arterial disease, and this is one of the earlier works where we looked at transfection of VEGF, vascular endothelial growth factor. And for this, we actually used a bicistronic vector where we tagged VEGF with GFP to allow for localization of where transfection is. And this is a rat model of chronic hindlimb ischemia where we ligate and remove a portion of the iliac artery, create chronic peripheral arterial disease. We deliver VEGF, EGFP by ultrasound-targeted micromobile instruction to the proximal hind limb, and then we look for bioeffects. And we have a couple of various control groups. We used a control vector that had no effect and also no therapy. You can see with the graph here showing transfection of the VEGF GFP bicistronic vector peaking at about day 3 after delivery. This is plasmids, peaks fairly early, and it wanes over 2 weeks after. So it results in targeted transfection, but it wanes out over time. You can see the green fluorescent protein in the vessels here. This is endothelial transfection. This is co-localization, DAPI nuclear staining here showing the vascular transfection. These are perfusion images. An example, control animal, just GFP alone, VEGF here. You can see the increased blood flow by contrast enhanced ultrasound in the treated animal and the reduced blood flow in the control animal. An example of filling curves on the right. This is fairly increased blood flow in the VEGF treated animal versus the 2 control animals where it remained quite ischemic. The graph here on the bottom left shows microvascular blood flow over time. In the white is day 28, so about 2 weeks after delivery. Let's go back. 2 weeks after delivery, you can see the almost doubling of blood flow to the ischemic hind limb in the VEGF-treated animals, and it does decrease over time, still significantly higher at 8 weeks, but definitely a fallout. And on the right is a fluorescent microangiography. It's a method of vascular casting, looking at the vessels. You can see the plethora of an increased vessel density within the VEGF-treated animal compared to a normal hind limb or to the ischemic hind limb treated with a GFP control. And you can see the waning of the vessels over time. And as a result of that, one of the next studies done by Alexandra Smith, who was a master's student in the lab, looked at ways to try and enhance the angiogenic effect in peripheral arterial disease and get that to stay longer, not have that effect over time. And what she did is that she combined a VEGF for early angiogenesis with another pro-angiogenic molecule, angiopoietin-1, which is involved later in the angiogenic process. And she compared VEGF to control, combination of VEGF and Ang1 delivered together, and then a combination of VEGF delivered early to have the new vessels grow, and then Ang1 late to prevent the new vessels from regressing over time. And you can see that the strategy of VEGF early and Ang1 late ended up with the highest degree of blood flow late after gene delivery, suggesting that multi-gene therapy potentially could be better than single gene therapy. And then she went on to investigate why that was the case, and she looked at pericytes. So pericytes are these cells that line vessels that provide the supporting structure for these vessels, allow these vessels to be maintained over time. And this is just some fluorescent immunohistochemistry where PGFR beta is a marker for these pericytes, showing that in ischemic animals, either control or VEGF alone, the pericyte coverage in these vessels is not as good as normal vessels, but with angiopoietin-1 late, you have really nice pericyte coverage. So a mechanism as to why these vessels stay patent for longer. We then went on to use other nucleic acids. This is microRNA, and we investigated a specific type of microRNA that belong to this family called angiomeres. They're identified in vascular endothelial cells. We focus on one called miR-126, and this is work done by Ron Wei, who is a master's student in the lab. And the way miR-126 works via the VEGF pathway, where it essentially blocks these two negative inhibitors of the VEGF pathway, SPRED2 and PIK3R2, and blocks these molecules to allow the VEGF pathway to work better. And this is some of Ron's work where he looked at transfection of miR-126. This is in situ hybridization images on the right, quantification here on the left with PCR showing day 1 and day 3 in the white and yellow bars, increased transfection of miR-126 after delivery. This is also another chronic hindlimb ischemia model in rats, and then the waning out at day 28. This is PCR in Westerns, showing the knockdown of the two inhibitors from a mechanistic standpoint, PIK3R2 on the left, SPRED1 on the right. These orange bars are the knockdown of these negative regulators of the VEGF pathway, and an effect that occurs early, wanes later. And this is the Western blotting shown on the bottom, just so you have both PCR and Western showing the effect of the knockdown of these negative regulators. This is the mechanistic aspect of research that we do on a regular basis. And with this is the biologic effect. This is perfusion imaging shown here on the top, contrast ultrasound. Red is higher blood flow, blue is lower blood flow. You can see that in the hind limb treated with miR-126, either single delivery or multiple deliveries, increased blood flow. The quantifications here are shown on the bottom, showing that miR-126 results in a doubling of blood flow compared to the two control groups over time. And then if you deliver multiple therapies, 3 in a row, separated by a day, you can actually increase or enhance that effect even more, just showing the possibilities of UTMD for pro-angiogenic therapies. Once again, this is fluorescent microangiography method of vascular casting showing the increased vessel density in the miR-126 chronic hindlimb ischemic model on the left, and this is the quantification graphs on the right showing the increased vessel density over time. So, some of the things that we've learned over the years for pro-angiogenic therapy for PAD, I think for clinicians, peripheral arterial disease, there definitely is a need for new therapies. It's a chronic problem, especially in diabetic patients. And multi-gene therapy or microRNAs or other non-coding RNAs really has good potential for this. And UTMD as a non-invasive method really has some advantages for repeat deliveries. I'm going to finish off with some of the work that we've done in acute myocardial infarction or ischemia reperfusion injury in the heart. So why target ischemia reperfusion injury? As a cardiologist, we see patients who present with acute myocardial infarction all the time. The primary goal is really to open up that occluded artery as quickly as possible. But we know that despite opening up that infarct-related artery, there's still an unmet need to further reduce infarct size. So therapies directed towards the myocardium that's ischemic, while opening up that infarct-related artery could potentially have some benefit over time. And we know that abnormalities of perfusion and metabolism are not restored in about half the cases after opening up that artery with a balloon and putting in a stent. And then reperfusion itself, that restoration of blood flow into the ischemic myocardium, is in part felt to be somewhat harmful. And it's estimated that about a half of the cell death that occurs during an infarction is caused by that restoration of blood flow. So how best can we protect that ischemic myocardium as we're trying to restore blood flow to create a smaller infarct size long-term, improve left ventricular function and better long-term outcome for our patients? So in collaboration with one of our investigators at St. Michael's, Tom Parker, who is an expert in S100 proteins, we looked at one of their specific proteins, S100A6. We know that it's a member of a superfamily of these EF hand calcium binding proteins that regulate a wide array of cellular and molecular function, including cell proliferation, calcium dynamics, cardiac contractility, and apoptosis. After a myocardial infarction, S100A6 is upregulated, expression is increased, And we also know that in transgenic mice that overexpress A6, they have less hypertrophy, they have less fibrosis, they have less apoptosis after ligation, so they are protected. So increased A6 is important for recovery after myocardial infarction. So Azadeh Mofid, who was a PhD student in the lab, studied A6 gene therapy using ultrasound-targeted microbubble destruction. And because at that time we were using mostly plasmids, we knew that transfection peaks at about day 3 after delivery. So what she decided to do initially as more of a proof of concept is to pre-deliver A6 to the myocardium in a model of LED ligation in rats. So she pre-delivered at day -2. She ligated the LED in animals 2 days later, and then we followed them over time. Because we knew that if we pre-delivered at 2 days before ligation, then peak transfection would occur around the same time as acute myocardial infarction, and we can have an idea of whether or not gene therapy is going to work. And she used a couple of different parameters to look at outcomes over time up to after 4 weeks after myocardial infarction. So this is some of her results. This is Mason's trichrome staining on the top, and you can see that the blue staining here is fibrosis. This is day 28. You can see the reduced fibrosis in the A6 treated animals, so a smaller myocardial infarction. This is tunnel staining for apoptosis, showing reduced apoptosis in the A6 animals early after a myocardial infarction. And she also looked at perfusion, these myocardial contrast echo images, where if it's this dark orange, there's reduced blood flow in control and null treated control groups, whereas A6 treated animals had more perfusion. This is LVEF quantification shown here on the right, showing in the orange line the increased contractility or ejection fraction in the treated animals, whereas it remained unchanged and reduced in the 2 control groups. This is apoptosis measurements showing the reduced apoptosis in the A6 treated animals, and this is myocardial blood flow, so increased blood flow to the heart treated with S100A6. That's a concept as to whether or not A6 potentially would be a potential therapeutic target to treat acute myocardial infarction. She then went on to use at that time a novel vector called mini-circle DNA. This is different from conventional plasmid vectors. They're smaller. They remove the bacterial backbone and transcription unit. They're supercoiled DNA molecules. They're smaller. And because of that, their transfection efficiency is greater. They result in much more rapid transfection and much longer transgene expression. And Azadeh used them in her experiments. This is just proof of concept in normal skeletal muscles, showing that with mini-circle GFP transfection as compared to conventional plasmid GFP results in more rapid transfection as quickly as 6 to 12 hours after delivery, and the quantification of transfection is significantly greater with mini-circle. So she ended up repeating her experiments, but instead of delivering them 2 days before, she delivered them really at the time of perfusion. So a bit of a tricky protocol where she did LED ligation and then 30 minutes after reperfused, and immediately after perfusion she delivered S100A6 mini-circle to the ischemic myocardium in these rats. This is PCR data, and she delivered human S100A6. You can see here the transfection of A6 within the infarct and peri-infarct zone as compared to the 2 different control groups that did not receive human A6. On the bottom is total A6, because rat A6 is produced endogenously after an acute myocardial infarction, once again showing the increased transfection levels if you have total A6, which is a combination of rat A6 and human A6 that we deliver exogenously. She used PCR to look at quantifying this. You can see the increased transfection in the infarct, peri-infarct, and non-infarct groups here with A6 mini-circle. And here's some of the functional data This is TTC staining. This is infarct staining in these rats. So white here is non-viable or infarct myocardium. You can see the smaller infarct size in S100A6 minicircle treated animals as compared to 2 control groups, either no treatment or treatment with an empty minicircle. This is myocardial perfusion as shown before, so increased blood flow quantification at the bottom. This is infarct size graph here for the TTC staining showing the smaller infarct size. And we used echocardiography for cardiac function. This is ejection fraction here with black being the A6 treated animals, blue and orange on the bottom are the 2 control groups. You can see the improved ejection fraction over time with A6 treatment and the fractional shortening. So just 2 different measures of left ventricular systolic function. Once again, Mason's trichrome staining. Blue here is fibrosis. You can see the reduced fibrosis or reduced infarct late at day 28 after ischemic perfusion or acute myocardial infarction. So this is still in manuscript preparation. So, what have we learned about ultrasound-targeted gene therapy for attenuation of ischemia reperfusion injury? Well, we know that contrast ultrasound has established and important clinical applications. I think I've shown you that. We know that nucleic acids can be bound onto carrying microbubbles delivered with a high-power ultrasound. You can target the genes at the time of reperfusion, and if you use a vector that transfects very, very quickly, you can actually have a fairly rapid biological effect even early after acute myocardial infarction. And at least A6 as a potential therapeutic target results in a smaller infarct size, improved systolic function and less fibrosis, and hopefully improved outcomes. So the question is whether we can translate this potentially into the clinic. There's this field of research that's ongoing currently called sonothrombolysis, where we try and enhance reperfusion with just regular microbubbles and ultrasound to improve outcomes after acute myocardial infarction in patients. This is one study published a couple of years ago where 100 patients were randomized to diagnostic ultrasound and commercially available microbubbles, so no gene therapy here, but just the biological effects of microbubbles and ultrasound to see if it can enhance the recovery after acute myocardial infarction versus just standard therapy with stents alone. Overall, it showed some promise, a trend towards improved LV systolic function with sonothrombolysis, smaller infarct size by MRI, and some ongoing studies to see whether or not this can translate to a biological effect. So, theoretically, if you can come up with a commercially available microbubble agent that can carry genes or other specific targets, you can deliver it at the time of reperfusion and further enhance this improved biological effect after acute myocardial infarction. So I'm going to finish. I want to thank you for your attention. This is knowledge. Folks in the lab, Mike Kolashevsky and Jim Saporis, have been in the lab for many years, kind of the backbones of the lab. This is a list of some of the master's and PhD students in the lab over the years. I've highlighted some of the work from Sandy Smith and Azadeh Mofid and Ron Wakow. And these are my key collaborators over the year. Jonathan Lindner has remained a collaborator. He was one of my supervisors when I did my research fellowship years and years ago. Tom Parker's lab is the expert in A6, and lots of other collaborators and friends and colleagues and mentors over the years, and then the various funding agencies. So thank you for your attention. Happy to take any questions. Dr. Martino?